Welcome everyone to the ChemInfo Stories webinar series. I am Ákos Tarcsay, the Product Manager of Calculators at ChemAxon. I want to share with you our recent project to extend the currently available training functionality of our calculators and introduce you our developments to exploit data with automated model building through machine learning. Machine learning, by definition, is giving computers the ability to learn without being explicitly programmed, and this is part of the core message of this talk. It is generally accepted that machine learning is to be applied when data is cheap and abundant, but knowledge is expensive and scarce. Think of applications like image recognition, self-driving cars, Google or Facebook scale of data. But in real-life drug discovery settings, collecting quality data is costly. Therefore, predictive models to help stratifying ideas or translating data into knowledge by deconvoluting relationships is highly valuable. ChemAxon develops calculators for 10 years to tackle this need and became a strong and recognized provider of cheminformatics toolkit, calculated physical-chemical properties and descriptors. To illustrate this with a real application, I cite here a Nature Reviews drug discovery study published 5 years ago by AstraZeneca, Eli Lilly, GSK and Pfizer scientists on analysis of large-scale clinical results. The analysis reaffirms that control of physical-chemical properties during compound optimization is beneficial in identifying compounds of candidate drug quality. These companies agreed on using ChemAxon calculators as on this invaluable dataset. To quickly show here a recent analysis of basic ionization constant prediction accuracy on publicly available AstraZeneca dataset deposited in Campbell, obtained with a general model. Considering the 1 log units pKa change correspond to only to 5.9 kJ per mole free energy change, these calculations provides a tool for active projects to balance attributes via predictions, since nearly 50% of the cases was predicted within 0.5 pKa units, and the root mean square error is 1.11, and the Pearson correlation was found to be 0.88. If we plot the experimental results versus the predicted values, it highlights that the general model yielded reasonable accuracy, but for unseen chemotypes originating from proprietary building blocks and novel chemistry, novel modalities, offset of the accuracy or accuracy drop might occur. This analysis, on one hand, inspires us to tune the general model further, but on the other hand, underlines the impact of the already available training functionality. When we are looking for future perspectives for improvements, 3 areas were mentioned by users. First, they want to use their collected physical-chemical data to augment predictions. Second, they want to use their data to build models for new targets. And third, due to the value of curated data and derived models on the field of generic targets related to ADMET and toxicology, predictions and predictors are still missing. Based on these observations, we decided to develop a new training functionality. We set 2 objectives. First, to fine-tune the trainable calculators and offer a more convenient interface to train and to run the trained models of pKa, lipophilicity, and solubility. And second, to build a module to predict molecular properties by actively learning from input data and provide models for novel endpoints. If we are looking at the generic user workflow in a high-level overview, it starts with collecting and curating the training data that can originate from in-house data warehouse or data sources, or from public resources like Campbell or PubChem. When this data is submitted to the training model, it will rely on our expertise in capturing the rich information of the molecular structure through our descriptors and high-quality physical-chemical predictors, and combining it with state-of-the-art machine learning methods to automatically build and validate models. This module will also serve the persistency and execution layer as well for novel series, novel molecules. The first interface is programmatic access through command line and REST, but we plan to release an intuitive graphical user interface with the application in the next stage. As a result, you can either get an augmented calculation for physical-chemical properties, including solubility, pKa, and lipophilicity, or new models, for instance, for ADME and toxicological endpoints, and as an example, I list here hERG, bioavailability, or your custom target. And this goes back to the history of this project, what we started with a large pharmaceutical company who shared a piece of their data to build the application, the Trainable module, and they have tested this in their pilot project. Highlighting some key technical points without going into the details. In terms of descriptors, we include both fingerprints and the wide array of molecular properties, plus utilize the flexibility provided by the ChemAxon toolkit to customize and optimize them. For featureization, we are investigating the effect of standardization of the input molecule, for example, in terms of neutralization, tautomer state selection, and aromaticity. In the future, we plan to enable using own descriptors or even previously measured data and combining it with our calculated descriptors. Related to model building, the current state enables regression models, and we are working on the classification type as well. From modeling approaches point of the view, we are experimenting with various techniques, including random forest gradient boosting, graph convolution, and support vector machines to find the best fit. The results collected so far indicates that hyperparameters for this life science use case can be pre-optimized. However, we are developing an on-the-fly grid search optimizer for the next stages of the tool to achieve automatic problem-tailored models in terms of the hyperparameters. Quality assessment was mentioned from two angles during the interviews with users. First, retrospective model validation and cross-validation to help triaging models before deploying it for larger scale productive usage. And second, information on the actual prediction by providing reliability estimation or applicability domain was also mentioned with similar crucial importance. Here, we plan to build error models and utilize conformal prediction framework that is recently getting more and more attention and more widely used and utilized in the industry. As an example, for today we selected the challenging hERG off-target endpoint that is related to cardiotoxicology. Hereby, I present preliminary results obtained with our training engine prototype. The training was done on a 6K dataset with the objective to predict hERG P activity values, and the original dataset was measured with electrophysiological assay. As you can see on this slide, the statistical parameters reveals that the model the obtained model has high quality. The root mean square error was found to be 43, the Pearson correlation is 0.75. 78% of the cases was predicted within 0.5 p activity units, and 98% was within 1 p activity unit. The test set size on which these results were measured is 1.5K, and you can see the distribution of the delta values from the experimental data that indicates that it is balanced towards lower and higher predictions as well. If we are analyzing the results further by plotting experimental values and predicted values on the test set that is derived randomly from the entire data pool, it shows that it is unbalanced towards lower activity ranges. However, it reflects typical drug discovery settings where early hERG screening is done in 10 or 1 micromolar concentration, which translates to 5 or 6 on these charts, and taking the desired 30 or 100 times selectivity window into account, this activity range is still reasonable. And it is important to be mentioned here that our random forest model for this use case picked with high relative importance complex descriptors related to ionization, microspecies distribution, average charge at a given pH, or lipophilicity. So if you ask as a user, what will I get as a result of this development? First of all, a new interface to train existing models for solubility, pKa, log-P, and log-D. Second, a new interface to train and run models for custom endpoints. And third, we want to ship ready-to-be-applied models for ADME and toxicological endpoints. And we are planning to make this utility available in ChemAxon Design Hub to offer a convenient graphical interface early on for testing and to embed this into a more elaborated design context. So in summary, these developments will ship access to extended training capability through our pipeline of feature engineering, machine learning, and model validation. If you are a medicinal chemist daily predicting properties to optimize the attributes of your ideas, or if you are a machine learning expert, a computational chemist, please contact us to get access to this prototype phase code, which is distributed in a Docker container for seamless deployment. We are open for early adopters who will shape the application together with us. Thank you for watching this webinar. Stay tuned for additional elements. Bye.